Good afternoon, children. Welcome to another practical assignment for today. Practical number 7. Today's date, 14th of August, on a new sheet in a corner. And I'll remind you once again the headings to be written in black pen. The aim of the experiment is study of osmosis through a Thistle funnel experiment. So what is Thistle funnel? It's a glass funnel which is made up of a structure like this and open at both the ends. So this broader end is to be tied up with a semi-permeable membrane in the experiment for today's study. Theoretically, I have explained this from page number 38 while we were discussing chapter 4 on absorption by roots, the processes involved. So in the assignment for today, children, you can see there are certain things that I have removed with the red pen and instead written down something else or added up some other things. So you will please write whatever I have written instead of what is cut here. The reason being that whenever the schools reopen, we will be conducting the experiment with the things that I have written here. So let's write what we will be performing in future. So the next heading, requirements, Thistle funnel, burette stand, beaker, 20% sugar solution, rubber solution, scissors, thread, marking pencil, water and cellophane membrane. So instead of writing these pig's bladder or parchment membrane, please cut this, we will write cellophane membrane. The third heading comes the procedure. Take a thistle funnel, close the wide mouth of the funnel with a cellophane membrane. Full stop. So all this is cut, children. This is not to be written till here. Continue with the second statement. The terminal edges of the membrane are then sealed by applying rubber solution. It makes the joint watertight. Pour 20% of sugar solution in the thistle funnel till it stands about 1/3 of the height of the stem. That means when you're adding the solution to the thistle funnel, it will be somewhere here around 1/3 of the height of stem. Now dip the thistle funnel in a beaker containing water. Support it in vertical position by means of a stand. So this stand you can see is a burette stand. Then your next point would be mark the initial level of sugar solution as A by means of a glass marking pencil. Till here is your procedure. Remember, children, this demarcation is your ending point of the heading procedure. After this starts your next heading as observation. I repeat, your heading starts here, the fourth heading, that is, first was the aim, second was requirements, third was procedure, which completed here. So the fourth heading, observation, begins here. So what we will observe? Note that after an hour, the level of the sugar solution has risen in the stem of thistle funnel to a point B, while the level of water falls down in the beaker. So after 1 hour, what are we going to see? That the level from A rises to level B. At the same time, the level in the beaker here would slightly, very slowly, and this will be falling down. The level in the beaker falls, but in the thistle funnel rises. Then, taste the water of the beaker. It is not sweet. Since the water is going inside, sugar molecules have not come outside, so the water in beaker will not taste sweet at all. Next heading would be conclusion. The rise of sugar solution in thistle funnel can only be due to the entry of water into it through the cellophane membrane. But no sugar has gone out into the water of the beaker as it does not taste sweet. The experiment therefore proves, number 1, cellophane membrane is a semi-permeable membrane because it allows water to pass through it. Second, sugar solution is an osmotically active solution and can absorb water when it is separated from it by a semi-permeable membrane. So, I hope this is clear till here. Continue next with the third point under conclusion. Water diffuses into a solution when the two are separated by a semi-permeable membrane. The phenomenon is called osmosis. Certain precautions are a must. First, the edges of the membrane must properly be sealed, right? Which edges we are talking of? When over this broader end of thistle funnel, when we are going to tie up a semi-permeable membrane, which is cellophane membrane in this case, so the edges should be tightly tied up. Then the thread should be tightly tied up and make sure that the edges are properly sealed. The initial level of sugar solution should be marked only after dipping the mouth of the thistle funnel inside the water of the beaker. And finally, fix the thistle funnel firmly in its vertical position by means of a stand. Right? So the working is simple, children. First, the setting up of apparatus has been explained, that how the thistle funnel has been filled up with 20% sugar solution. Then the semi-permeable membrane, cellophane membrane has been added, tied up. Then the edges have been sealed up and the agent used is rubber solution. So doing this, it becomes airtight, and now we will dip the setup in a beaker containing water. What we observe after 1 hour is that the level here that was marked earlier as A has risen to B because of osmosis. So this is what you're going to study through the thistle funnel experiment today, right? So I'm going to send you the writing part, children. It begins from here, all this. Make sure, please stick to all the changes that have been made in red pen. The remarkable change is here, that the procedure ends up over here and the observation begins. So your writing part is very simple. Diagram would be like this. Label it clearly, children, and your ending part would be at precautions. So that's all for just now. This will be your practical number 7, and your submission date would be by 17th of August, because there by evening I'll give you, or maybe by any other time I'll post the practical number 8. That's all for just now. Thank you.